Alright, so in this lecture we're going to look at the genetics of blood types. So very few in human beings, we're finding that less and less of our traits are actually controlled by single genes. Most of our genes our genetics are polygenetic traits, which means that there are a number of genes that control each thing. And the easy way to tell if something is a single gene trait or polygenetic is like if it's all or nothing, it's a single gene trait. Okay, if it's kind of a blend, then it's a polygenetic thing, like height. You know, there aren't just two heights. There aren't just like tall people and short people. There's like every imaginable variation in between tall and short. That's a polygenetic trait. Okay. Skin color is a polygenetic trait. There's every imaginable variation between light and dark and things like that. Okay, but blood type is one of those things that is controlled by single gene traits. So like basically there are multiple alleles for blood type. That will determine well, blood type. I guess that's kind of redundant. Maybe I should have thought that through. So like if I've got a couple of chromosomes here, and I'm going to just draw them kind of simple like that, a couple of chromosomes, and there's a gene on those chromosomes for blood type, and I really off the top of my head don't know which gene, like which chromosome that's on. Gene for blood type. And these are supposed to represent chromosomes. Well, we have two copies of them. You get one copy from mom and one copy from dad. Okay. The alleles or variations of those genes include big IA, which is dominant for A, big IB, which is dominant for B, and little I, which is type O. Okay. So let's look at some genotypes and phenotypes. And if you remember back to biology, I will also say that blood type, oh yeah, there should be an S there. That blood type is an example of codominance, where you can have more than one dominant allele at a time. Phenotypes and genotypes. Phenotypes, basically what's expressed, what does it look like, and genotypes are what are the genes that represent that. So for type A blood, you can have big IA, where you have, that's homozygous dominant for A, or you can have big IA, little I, which would be heterozygous. You can have type B, which would be big IB, big IB, big IB, and little I, which would be the heterozygous version of this. For type AB, you can probably guess, you're going to have big IA and big IB. And for type O, you're going to have little I, little I. That is the only possible outcome for that. Okay. So, let's just look at one of these here, an Punnett square. And so I'm going to choose the one that would create the most variance. And I actually had a student, actually no, I had 4 students, 4 kids from the same family, and we got to type all of their blood types over the course of a number of years. And here's the story, okay? Mom or dad, I don't remember which one, was A, but they were heterozygous, so they were big IA, little I. The other parent was big IB, little I. Okay, so let's make a Punnett square here to see how this plays out. Now one goes on the top, one goes on the side. It doesn't matter which one's which. 4 times 5 is 20. 5 times 4 is 20. You end up with the same result regardless of who goes where. Some say, oh, the male's supposed to go here, the female's supposed to go here. Don't follow that logic, because it breaks down with plants, because flowering plants are both male and female, so that doesn't work. Big IA, little I. So this first one would be big IA, big IB. Next one, big IA, little I, big IB, little I, little I, little I. So in this case, you'd end up with 25% big IA, big IB, and that would be type AB, 25% big IA, little I, type A, 25% big IB, little I, type B, and 25% little I, type O. And I don't know if I told you or not, but when we typed the blood of each of these kids, every one of them had a different blood type. Now, it's not to say that it will always work out that way. It's saying percentage-wise, you have a 25% chance. All of them could have turned out type AB, but it just happened that their family was a perfect Punnett square for this here. Now, there's a number of different things we can try here. We can do O, but if you do all O, we would have little I times little I, and you'd end up with all little I. So there's some things that are possible, there's some things that are not possible, which is why one of the reasons I have a permission slip with my blood typing lab, because in rare instances students can find out they're adopted if both their parents are O and they show up as anything other than O. They may find out something that they weren't prepared to find out. So but there are cases like, let's say both parents are A. Well, if both parents are heterozygous for A, there is a possibility that their kids, 25% chance that the kids could be O because of that recessive allele that's hanging out there. So that's the gist of it. Basically, you take whatever the genotype of each parent and you plug it into the Punnett square and you find out what the percent chance that each of the offspring could potentially have of each type of blood.